There are two kinds of pressure that we have to consider when we're talking about blood pressure. There's a hydrostatic pressure, which is the pressure of water, kind of like if you had a water balloon, the pressure that the water exerts from the inside onto the balloon is that hydrostatic pressure. You fill the balloon up more, you get more pressure. But there's also something called colloid osmotic pressure, and this also makes a difference as to how much pressure is in your um, vascular system. A colloid, it's just a substance in water, and it's a substance that attracts water. So, like any of the solids, red blood cells, white blood cells, any of the ions, right? Anything that can attract water is a colloid. Colloids attract water. And we measure colloids in your body by looking at the blood colloid osmolarity, or we can look at the colloid osmotic pressure. They're all the same thing. Usually on a CBC, you see it measured as osmolarity. All right, so let's say that you have one of these luscious plants here that I could kill in a second. And, the plant starts looking bad. So what you do is you give it some fertilizer. And fertilizer, actually, it has a lot of solutes in it. It does. Like, that's the purpose. It's got nitrogen, it's got other things that the plant can use. And so now you have a salty plant. Now you have a plant living in really salty soil, right, which is not good. And then you water the plant because you're like, oh, now it looks even worse that I fertilized it. Let me water it. And what happens is all of the water just sticks to the colloids. That's it, right? The water is attracted to the colloids that are in the soil, and it never actually gets into the plant. So don't be like me. Don't fertilize then water. You're just killing your plant twice. In the picture below, we have a capillary bed being fed by an arteriole and drained by a venule. In the capillary bed, the hydrostatic pressure, or the pressure of the water, pushes water and solutes out of the capillary bed and into the surrounding tissues. The colloid osmotic pressure attracts that water back into. So there are two kinds of exchange here. There's filtration, which is this arrow pushing solutes and water from the capillary to the surrounding tissues. And then there's reabsorption, which is mostly just the reabsorption of water. And this reabsorption represents the attraction back into the capillary of those lost fluids. Not all of them, but most of it. This diagram, it also shows a capillary with the surrounding tissue cells and open space between the capillary and the tissue cells. So don't forget that you have the intracellular fluids in these cells that I'm drawing here, but anything leaving the capillary would have to go through the interstitial fluids in order to get to the intracellular fluid. So filtration is depicted on the left side of this capillary bed, the arterial side. And these arrows show substances moving from the capillary to the interstitial fluid and ultimately into the intracellular fluids of the tissues. Filtration is movement of plasma components, things found in the plasma of whole blood, into interstitial fluids. Filtration pushes water out. Most of the larger solutes stay in, like red blood cells and white blood cells, but most solutes that are small, like glucose and amino acids, they escape the capillary by way of filtration. And this process is driven by the hydrostatic pressure of water, right? Filtration is guided or caused by that hydrostatic pressure. Reabsorption, on the other hand, is shown on these arrows on the right side of the diagram, representing substances moving from the interstitial fluids back into the plasma. And this is when mostly fluid moves back into the capillary, and water is attracted to all the solutes that are inside the capillary. And so reabsorption is driven by the colloid osmotic pressure. There's a diagram in your book that I just feel like it makes this more complicated than it has to be. But what it has is it has blood entering into a capillary bed from an arteriole, right? And we know that this here is a venule that is draining this capillary bed. As blood enters into the capillary bed, filtration occurs, right? Like immediately there's stuff that's leaving the capillary bed. It's like, oh, I'm headed for the interstitial fluid and the cells that are in there. Later, reabsorption takes place as substances move back into the capillary bed. And we know this happens on the venule side, right? And this is fluid moving from the interstitial fluid into the plasma. So this movement creates what is called a net filtration pressure. The difference between filtration and reabsorption pressures. The difference between the hydrostatic pressure that makes filtration and the colloid osmotic pressure that makes reabsorption. And the only thing that you need to know, despite all of the numbers that your book uses, is that filtration always exceeds reabsorption. All of the fluid that comes out by way of filtration is not reabsorbed on the other side of the capillary bed. The excess fluids are managed by this structure here, which are lymphatics. Lymphatics are like a drainage system. They collect all of this fluid from all of your tissues and they dump it back into the subclavian vein. So it's a drainage system. It's also able to take in large things like white blood cells and proteins, which the venule side really doesn't do. It takes in smaller things. Lymphatics also, and this excess fluid that we're talking about here, can be the source of edema. When lymphatics fail to drain the excess from a tissue, fluid builds up between the cells of the tissue and it causes edema. We always forget why it is that we're talking about blood pressure. And we're talking about it because filtration and reabsorption are critical for tissue perfusion, right? The exchange of oxygen, glucose, and water into the tissues. I want blood to flow. I want resistance to be low. I want blood pressure to be low so that I can get good tissue perfusion, right? And it ensures that I'm able to drop off oxygen, take up carbon dioxide, distribute hormones, and remove toxins and pathogens, right? A common issue arising from the imbalance of these fluids is edema within a compartment. And this is caused by those excess fluids. And it can result from increased vessel permeability of your lymphatics. And it can also result from a low blood colloid osmotic pressure. Like if you have low proteins in your blood, you can also experience edema. So it's not just the lymphatics, it's also the colloids in your blood. When colloids in your blood, like if you're low on plasma proteins, which you can be from malnutrition, when colloids are insufficient, water can't be reclaimed through reabsorption. And it also has issues going into the lymphatic system. The lymphatic system also needs proteins to absorb excess fluids. So this leads to swelling. And this is also what accounts for the massive swelling in the belly with starvation. So bruising is a local form of edema. Starvation can cause ascites and other edemas in the gut. And high blood pressure can actually exacerbate these conditions. So prolong these issues, right, for years, not paying attention to them, not going to the doctor, what it ends up doing is it ends up in peripheral congestion. Little by little, more and more blood ends up like staying in your systemic circuit and not filtering back and forth through the pulmonary circuit. So little by little, more blood ends up congesting your peripheral circuit.